Vijay, please go ahead.
Thank you. So next up, we have Dr. Kumar Kothapalli, who is an associate professor of pediatrics at Dell Medical School, UT Austin. He is well known for his work on Uddanam Kidney Disease and has published several papers in well-known medical journals. He will be presenting on inherited thrombophilia. Please start sharing your screen.
Thanks so much, UPASA and Gaman Foundation for giving me this opportunity. As Neha mentioned, Neha and Vijay has done a great job in organizing this, and I thank even Roja Garu, mother of Vijay, for organizing at the back end, and she has supported so much for this event. I really thank executive team of UPASA and executive team of Gaman for bringing this to the worldwide audience. So today, my topic is in genetics. So it's completely new to most of the kids here, because the experienced doctors might have heard about the inherited thrombophilia. So I'm an associate professor at Dell Med, but this talk is only for awareness event. So it's nothing to do with my medical school. And I'm presenting on behalf of UPASA. So if we see in genetics, every nucleated cell have thread-like structures which carry several genes. And in human, if it is 46XY or XX, we are normal. And then when we see these chromosomes and when we thread-like structures, we have a DNA, which is the genetic material made up of 4 nucleotide bases: adenine, cytosine, thymine, and guanine. The human total DNA is like 3 billion bases. And gene is a discrete part of DNA, which makes specific proteins. And in humans, we have 20,000 protein coding genes. And we say that the DNA has 3 billion bases, but what happens if there is a mismatch in any part of our gene? What it causes? It can cause either inherited genetic diseases, and some cases, and especially in some specialized conditions like cancers, we see a lot of somatic variations. Inherited mutations, they pass on to next generation, but acquired somatic mutations, they are only accumulated during a person's lifetime. They are not passed to the next generation. So we need to be very specific here. Inherited mutations, they pass to next generation because they are in the germline, whereas somatic mutations, they occur during a person's lifetime, so they are not passed to the next generation. Especially 90% of cancers now are somatically driven. And thrombotic risk is very high in these cancer syndromes, cancers. Let us take, like, if we have like 3 billion base pairs, what happens if there is a change in one DNA base? If you see here, this A is adenine in normal patients, in normal individuals, whereas if it changes to thymine, we see a sickle cell anemia. So during my talk, I represent this, image as normal. When we have a one copy, abnormal copy from one of the parent, it's a heterozygote. When we have 2 copies, it's a homozygous condition. Generally, homozygous conditions are very severe. We will see very severe phenotypes. If you see here in sickle cell anemia, you see a anemic, or in normal we see donut-shaped hemoglobin, whereas here it's a sickle-shaped, because change in one nucleotide in among the 3 billion base pairs of human DNA. Today, I'm going to talk about only the inherited part, not the somatic part. So if we see here, there are 30 genes in the coagulation cascade, but well studied among them are factor 5, factor 2, methylene tetrahydrofolate reductase, and PIA1. Whereas the JAK2 is related to acquired mutation, and it is a hallmark for myeloproliferative syndromes. Among the most common inherited thrombophilias, factor V Leiden, prothrombin, and elevated homocysteine levels are the major things. Even we see a lot of protein C and proteinase deficiency, and some of the gene defects in those genes are also there, but well studied among them are factor V laden and prothrombin gene mutations. So why we want to see factor V in highly hypercoagulable conditions like COVID? Because if we have a heterozygous genotype in factor V, there is a lifetime increased chance of 4 to 7-fold in a lifetime, getting a thrombotic event. If you are a homozygous, then the chances are 50 to 80-fold. So if you see the incidence of factor V, especially in European whites and United States, it's very high, especially, this 2 to 15% are heterozygotes, whereas homozygous factor V Leiden mutations are extremely rare. But when in hypercoagulable conditions, we need to see that, because they're having a second hit, then, these individuals might be more prone to having more thrombotic episodes in their lifetime. Especially, we need to be very careful with the homozygous individuals because they are at very high risk. Let us see here, the Europeans, we see very high incidence, especially if you see here, the Spain, France, Germany, Iceland, UK, they are all having very high incidence of this factor V Leiden mutation. When coming to India, we have only 1.3 to 4.1%. That means when compared to the Caucasian populations, Indians have very less incidence of factor V Leiden defect in our genomes. And if you see in Japan, it's completely absent. It's very important to have this mutation checked, especially when we are seeing persistence or, when there is Dr. Vijay Kumar Garu has presented, like in Netherlands, we see like even after anticoagulant therapy, they are still coming back. Why? Because this factor V Leiden has been discovered in Netherlands, and the incidence of this mutation in European population is high. Hello. Yeah. So especially when we are using oral contraceptives, and if there is a heterozygosity involved, then the increased risk for thrombosis increases. And especially in homozygous condition plus oral contraceptives, the fold increases very high. We need to be extremely careful when we see, like Dr. Nagreddy Garu said, especially when we are prescribing oral contraceptives, and if they are having thrombotic episodes, we need to suspect, especially in the Caucasian population where the incidence is high, we need to suspect, like, that factor V could be one of the reasons for having these, episodes. And factor V accounts for 90 to 95 cases of activated protein C resistance. And if there is obesity, smoking, and old age, the risk increases in heterozygotes and homozygotes. Especially cancer is a hypercoagulable condition. And there is a one case control study from Dana-Farber showing that women who have on tamoxifen citrate, their chances of thromboembolic events are 5 times more likely if they carry a factor V Leiden mutation. So this is my I cite from my published last week, where we have shown, like, inherited genetic risk factors can enhance or have an additive effect in increasing the risk of thrombotic events during hypercoagulable periods such as severe COVID. For example, if there is a patient where they carry mutation in both factor V as well as in prothrombin, their chances of having a thrombotic risk is extremely high. So we need to be very careful with the patients who are homozygous for factor V and who are homozygous for prothrombin factor II. They have a very high chance. So if any patient who carries this factor V in a heterozygous condition as well as prothrombin in a heterozygous condition, the chances of having the risk of thrombosis is very, very, very high. And there is one paper showing like patients with factor V mutation displayed high levels of D-dimer and fibrinogen fibrin degradation products in plasma. And if we see the incidence of a factor II genetic defect or a mutation in population worldwide, again, we see the high incidence in European Caucasians. And if you see the incidence in Asians, it's completely zero. And in India, it's one paper has shown from 0 to 0.6%. But from my experience, seeing a lot of genetic cases in India, we have never found a factor II G20210A mutation in Indian population. I haven't seen, I have given several talks, and from my experience with my colleagues in India, we haven't seen prothrombin gene mutation in Indian population. We have seen in this mutation in Hispanics, African Americans, and especially in very high incidence in Caucasian, some of the Caucasian populations. And when you see the COVID, especially in Italy, Spain, and France, the death rates were very high during the initial March period. Are we missing anything here? Are we missing any inherited thrombophilia conditions? We need to check. The take-home message is, what I call for young physicians here who are more challenging to take up, the cases and study even post-COVID. Please look into the incidence of inherited thrombophilias and see, what we can do in helping these patients. Methylene tetrahydrofolate defect is very common in 1990s, especially when it started in Netherlands. They investigated the MTHFR related to neural tube defects and like spina bifida and anencephaly. During that time, the folic acid was not that popular, and they were all naive population. So when we did the biochemical phenotypes, we have seen in heterozygotes also, there was elevation in homocysteine levels. If you see the CC normal, the homocysteine levels were less, whereas in heterozygotes, the homocysteine levels were high. But now we find folic acid everywhere. In US, we have folic acid in cereals, what we eat. So biochemical phenotypes can be misleading because we are exposed to folic acid now. The biochemical phenotypes can show lower homocysteine levels, but genetic phenotype remains the same throughout the person's life. So always we need to see how genetic phenotype, if we see a TT genotype, and if the homocysteine levels are low, we need to ask the patient whether he is on folic acid therapy or B6, B12 therapy. These conditions can lower your homocysteine levels. Especially Shruti Akula has presented on CBS mutation. CBS, when you have a CBS cystathionine beta synthase pathway deficiency, we see more high levels of homocysteine, and this causes thrombotic episodes. And certain cohorts in India, especially Kshatriya population, they have very high levels of homocysteine. This I have a conversation with a very well-known cardiologist in India, Somaraj Garu, and his colleagues. And we thought of like seeing, how we can do genetic screening or testing in India to identify these kind of defects, which can prevent long-term effects. So when we see homocysteine levels low and TT genotype, then we need to go for, ask the patient not to stop the therapies. They need to take it throughout probably, their lifetime, or based on the medical faculty who is advising the patient. And I thank everyone for giving me this opportunity. I thank Nandakishore Garu for organizing this event in India and collaborating with us. We are doing this thrombotic, thrombosis awareness events since 2015. In 2016, we did it in New Jersey at Dr. Sai Kolagari's office. Then I moved to Austin, Texas, and we started this chapter in 2018, and it's going on well. Thank you so much.
Thank you so much, Dr. Kothapalli, for that very interesting talk. The final delegate in this phenomenal panel is Dr. Nandakishore Yaramseti, who's a leading consultant and licensed physiotherapist. His expertise is in treating palliative, pediatric, adolescent, adult,